Today I'm going to talk about some ideas that I've been working on over the last year as part of my postdoc position, and just sort of exploring the potential for developing a metabolic theory that can explain questions of interest to community ecology, especially in microorganisms. And I just wanted to say a little bit about myself. So I graduated last year. I was a student of Jim Brown, and am now at the School of Earth and Space Exploration at Arizona State University on a fellowship there, working with Everett Schock and Jason Raymond. So metabolism is the uptake, transformation, and allocation of energy and materials. It is constrained by fundamental laws of physics and chemistry. A metabolic theory of ecology, then, seeks to try and understand the role of metabolism in ecological patterns and processes. And more specifically, a metabolic theory of community ecology looks at how metabolism might govern patterns in community structure, composition, diversity, dynamics, species interactions. Now, to gauge the usefulness of a metabolic theory of community ecology, one element is how many different kinds of patterns or phenomena the theory can explain with as few assumptions as possible. So, ideally, such a theory would explain as much of the biodiversity as it can. And if we look at a tree of life, we see that bacteria and archaea make up a large portion of life's phylogenetic diversity. Likewise, they exhibit a diversity of niches along different gradients in environmental or physical or chemical variables, such as temperature or pH. Oh, this thing is not. Let's see if we can get rid of that. And microbes also have the greatest diversity of major metabolic lifestyles. So in terms of their energy sources and carbon sources, they can attain energy from light, from inorganic chemicals or organic carbon, and carbon from carbon dioxide or organic compounds, whereas plants and animals occupy only two of these metabolic lifestyles. So given this remarkable diversity, there's a lot of potential insight that can be obtained by developing a theory that applies to all domains of life and addresses microbial diversity across extreme ranges of habitats that microbes occupy. I think my title should be cut off. Oh, pull it down. Oh, yeah. So,
there we go.
Move it out of the corner so
there, that should be
okay. This is a PDF. So if the first step in developing a metabolic theory of community ecology for microorganisms, I think is we need to think about the different aspects, fundamental physical and chemical aspects of metabolism. And a lot of the sort of formal or formalized metabolic theory is focused on chemical kinetics, which is basically understanding the rates of reactions. And we also need to understand thermodynamics, the spontaneity and direction of a reaction or process. So in this figure here, we can see that this is sort of like a potential energy landscape. So on the y-axis, the Gibbs free energy, which is a form of potential energy. And if we have a reaction, it's, in order for it to move forward along this reaction coordinate, it has to go up this hill, which is the activation energy E. And the height of that hill, that initial hill, affects the rate of the reaction. Now, when it gets down here, the change in its potential energy, that reflects the total amount of energy that's basically released from this reaction. So that's the delta G, the change in the Gibbs free energy. For a reaction to be spontaneous in this forward direction, the delta G would be negative, and where the value, the absolute value of the delta G represents the energy yield of the reaction. And in order for there to be an energy yield for that reaction, the reaction has to be in non-equilibrium thermodynamics, thermodynamic non-equilibrium. So the greater the non-equilibrium, the more energy that's yielded by the reaction. Now we can see that the metabolic rate, which is power, it's energy per unit time of an organism, it represents both the delta G as well as the rate of the reaction. So where R is the rate of the reaction, delta G here. So the summation of, or the multiplication of the rate of the reaction times the amount of energy yielded by the reaction gives power of that reaction. And then organisms' metabolic rate, or in ecosystems, is going to reflect the summation of all those different reactions. A variety of work has been done in biology and ecology showing how temperature affects reaction rates, and often it can be well approximated by this Boltzmann factor here. Of course, though, at extreme hot temperatures, this approximation fails because for enzyme catalyzed reactions, so for biological reactions, because the enzymes become denatured. And this can be especially important in microbial communities where there are a lot of microbes that are living across a wide temperature range. Now, there are models that deal with this denaturation process and how that would affect reaction rates, and that requires integrating sort of thermodynamic and kinetic effects on the enzyme stability and ability to catalyze the reaction. What I'm going to focus on a little bit more today is the delta G and its role and potentially its usefulness in community ecology of microbes. So at the very simple level, delta G is this equation here, where K is the equilibrium constant and Q is what's called the activity product. I won't go into those here. But suffice it to say that when this is actually applied to a sort of the real world or a natural system, that being anything beyond a very simple laboratory system or sort of thought experiment, there's actually, the equation can be kind of misleadingly simple, as in, for example, temperature can affect K and Q as well. So it's not necessarily just a linear relationship between delta G and temperature. So what are the factors affecting this delta G, and how much does it vary? Well, it varies by 2 orders of magnitude, at least, for biological usable reactions. It can switch sign often along environmental gradients, and so that affects whether or not a reaction can actually yield energy that would be of use to an organism. So if the delta G is 0, or then that is no longer, that reaction is no longer a source of food, basically, of energy for an organism. Now delta G is affected by temperature. Like I said, it's also affected a lot by pH, the reactant and product concentrations, pressure, the ionic strength of the solution, for example, salinity, and in particular, pH impacts heavily the chemical composition of an ecosystem, as a consequence affecting concentrations of reactants and products, which then affects the delta G's of ecosystems. It also, pH will also can directly affect the reactions that involve hydrogen in them, which there are a lot of biogeochemical reactions with hydrogen. Temperature has complex effects on delta G, although it can, all else being equal, will have a linear effect on delta G. It tends to be a less important driver across a wide environmental gradients because of these other because of pH having such a strong effect. But if you hold pH constant, then the effects of temperature can emerge. Some of the stuff I'm working on is trying to understand what general ways does energy yield vary with temperature, pH, concentration of reactants for biologically relevant ranges. Now, geochemists and physical chemists who study this use a complex set of models and algorithms to calculate the delta G's based on the conditions that the reaction is in. But the question is are there any sort of emergent, more general quantitative effects that reflecting the underlying thermodynamic equations, whereas maybe in some cases there are idiosyncrasies that we can't actually come up with general predictions for. So I've been working on developing some mathematical theory to predict some of the general relationships for the energy yield of reactions. So it's sort of a macro of biogeochemistry. What I found is that there are linear effects of pH in many cases, and that can reflect the underlying thermodynamic equations. Everett Schock pointed this out in a 2010 paper, but didn't go into deriving some of the theory behind some of these patterns. As we see, sometimes pH does affect strongly the energy yield of a reaction, whereas other times it doesn't have as much of an effect. And so for certain reactions, it has a very strong effect. For example, this is a reaction from magnetite to iron with these other compounds, chemical species involved. And here affinity is basically the same thing as energy yield as I had before. So it's just another term used in that literature that reflects the energy yield of a reaction. And if the affinity is negative, that means that the reaction can yield energy, but in the backwards direction, basically, that it's written. So along a pH gradient, an organism might be able to attain energy from a reaction going forward, and then if at a higher pH, it would have to use the reaction in the backward direction, if it can, in order to obtain energy from that reaction. Now, temperature. So all else being equal, there are these linear effects, which we can see here for the case of a nitrification reaction. And this is plotted for calculations for energy yields for hot springs at Yellowstone. And it's along a runoff from a hot spring, so where temperature is changing, but pH isn't changing very much, and the underlying geochemistry isn't changing as much, which is perhaps why there's a more clear relationship than often is observed. So sometimes there's a lot of complicating effects when looking at how temperature affects energy yields, because temperature influences the amount of gases or chemical species that are in the solution, which in turn then affects the delta G. So, given these effects of these different environmental factors on delta G, and that delta G varies a lot and determines basically the quality of the energy sources for microorganisms and what energy sources are available, seems like we might be able to start developing assembly rules for microbial communities based on these thermodynamic principles. What the thermodynamics can tell us is it provides a first sort of constraints, absolute constraint on what biogeochemical reactions or metabolic pathways can be used in an ecosystem. So it can then be used to make predictions about biogeochemical richness, the number of biogeochemical reactions in ecosystem composition, and predict macroecological patterns of metabolic diversity, which imposes constraints on the assembly then of species or taxa, because different taxa require different reactions for their energy sources, and often some of these metabolic pathways are phylogenetically conserved. This is especially relevant for chemotrophs that they're obtaining their energy from geochemical reactions, whereas photoautotrophic organisms, this sort of approach with delta G's is maybe not quite as useful. Let's see. So, although the thermodynamics can be useful, it also has to be integrated with kinetics in order to get more precise predictions, because the rates of reactions are going to vary a lot as well, and that's going to determine as well that is going to contribute to the total power supply available to an organism and whether or not it can make a living in a certain habitat. And so here we see that in order for a species to be present in a given location, the power supply available to it, sort of its resource availability, R, has to be greater than its metabolic rates. How much time do we have? Oh, okay. And so, let's see, I basically talked about that. And so the gist basically of this is then we want to try and scale this up to make predictions about what species are where and how that might affect then the total species richness at different places. And basically based on these sort of underlying metabolic effects of temperature and pH. And and we can start to look at diversity gradients, for example, that might shed light on different hypotheses for understanding the metabolic basis of the latitudinal diversity gradient. So looking at species richness with respect to temperature in different communities. And as well as looking at how species richness changes along pH gradients. So to conclude, I think that it's important that for microbial communities to try and integrate the effects of thermodynamics and kinetics. And this is, in particular, it will help in terms of understanding how pH and temperature affect microbial communities. And I think also there's a need for collaboration between community ecologists and microbial ecologists, because often the two fields are sort of separate. So I think there's a lot of exciting opportunities for trying to integrate these across the three domains of life. All right. These are some people I want to thank for their advice and help with data as well, and also my funding support from ASU. All right, thank you.
Thank you, George.